Hello everyone, and welcome to our presentation on advanced and non-invasive glucose monitoring. The previous device, GlucoCheck, measured glucose levels in the blood in a non-invasive fashion, and that was done through a laser going through the belly of the finger, and then a camera takes the picture from the other side to pass it to the model to make the right prediction. Now we're trying to include physical factors like humidity on the skin, thickness of the skin, temperature, and skin tone, and see how these affect the accuracy of our current model and how we can include them or exclude them if they're irrelevant in the future development of the next deep learning model. So the skin factor, the physical factors are going to be skin humidity, thickness, temperature, and color. And the first study about skin thickness variable had the experiment done with 40 volunteers with two wavelengths, 635 and 805. And the lasers were shot at the level of a site near the elbow, right? So two variables were measured, transmittance and reflectance, which are the inverse of each other. In our case, we want as much transmittance as possible through the first layer of skin, the epidermis, and that way we can get most of the light in the parts where we have blood, red blood cells, proteins, lipids, and the most interesting molecule for us, which is glucose. Now, the effects of skin tone and tissue thickness were correlated to each other. So these two variables right here, they found that at 5 millimeter skin thickness, which is the higher thickness of the spectrum here, they noticed that differences in transmittance became insignificant, meaning that unless you use an 805 wavelength, the transmittance is going to be both the same for different skin tones. So you get high transmittance with a higher wavelength. The second study took care of skin humidity, in which they had this setting right here. Instead of using human skin, they used an agar phantom, which is a skin mimic, really used in the research area where you try to not use any animal or human subjects in the experimentation. So they had cryogen spurts being shot at the agar phantom and then a laser on top, and then they're measuring the transmittance. They noticed a 54 decrease of transmittance because of the cryogen film, because it reflects the light pretty much, and that's not what we want. We want as much transmittance as possible for the experiment to be for the measuring or for the image to be taken in a very natural manner. And then the temperature effects, we're trying to see like regular glucometers, those that prick the finger, and see if temperature affects them. And in fact, studies have found that there's a difference, statistical a significant statistical difference between warm and cold conditions, and that is caused by lower blood flow when you get your limbs colder, basically. So that causes some sort of a latency in measuring the glucose, which means we might have to put limitations on which temperature is it impossible for GlucoCheck or any glucometer for that matter to make accurate and reliable glucose measurements. Finally, we're studying the difference between using lasers as a light source and camera as a receiver. We're seeing here some potential in using an LED light, which is safer and even using a higher wavelength, and a photodiode that takes or receives light and converts it to a current. So a study done at the University of Qom in Iran, they found that there is a 12.33% error in measuring glucose, and the higher the glucose value is, the less that error is, meaning it's very reliable for people with diabetes to be careful with higher numbers. So the next step in our research is using a 1600 nanometer LED light combined with photodiodes, and see or replicate these results and see what we can include from the physical factors to make a more informed and reliable machine learning model to take care of this of this operation. The data flow diagram is we're using Qualtrics exports, which is a software to do the data collection on the day of it, and then using ETL processes, the data is being sent to influx database, and we'll get two types of data points. One is with photodiode voltage or current, and an IR infrared image, and potential features include both physical factors. And finally, the reference value is the blood draw glucose measurement. This research was partially funded by the Asian Research Program funded by the National Institute of Asian, NIA, part of the National Institute of Health, the NIH. Thank you very much for your attention, and we look forward to the day of the presentation, and good luck for everyone, and thank you for watching.